transfer student from the Cayman Islands. And,
I did not know.
We send students there every spring as part of our microbiology thing, but she gets to be there all the time. So she did, I did not have her for organic, unfortunately, and so I was really happy to see her completely crushed biochemistry and really fit in well here and doing a great job. She's going to talk about work that she did at Indiana with another, if you recognize Alex Hurlbut on that list, he is another Lawrence graduate from 2 years ago. And she says that she worked next to Konstantinos, who is also in that. Okay.
So hey guys, thank you for taking the time to come to my talk today, which is titled Elucidating the Levels of the Estrogen-Related Receptor of Drosophila melanogaster during Larval Development. And for those of you who may not be familiar with Drosophila, it's actually the fly that you see buzzing around the food section in the commons. So why did we use a fruit fly versus any other organism? Well, because it's incredibly easy to grow in lab. Their larval stages are relatively short, and once they reach adulthood, they reproduce really fast. Their metabolic processes are pretty similar to those in humans. Though humans are more complicated, it's a good way to start if you want to model, which is exactly what my lab was aiming to do. They wanted to create a model system to study aerobic glycolysis so that if anyone else is studying aerobic glycolysis in a more complicated organism, they can start with this model. And so the goal of my project is to find out the importance of ERR in metabolism, and we know that ERR acts as a switch that initiates glycolysis, but we don't really know the factors that actually affect it and how important or how vital it is to actually start in glycolysis. So what is glycolysis? Well, it's the breakdown of glucose and the first stage of energy generation, which can then go into other processes to create even more energy in, for example, respiration or fermentation. Glycolysis is important for rapid cell growth and development, not only because it creates energy, but also because some molecules that are created when glucose is being broken down can be used to make amino acids, nucleotides, and fatty acids. So just to make this more clear, in glycolysis we start with glucose and then it's converted to pyruvate, which is the end product made in glycolysis. And then pyruvate can go on to make acetyl-CoA, which can go into respiration, for example, or pyruvate can take a different pathway and be created into alanine, which is an amino acid that's essential for making proteins. And these are only two pathways of the possible pathways that pyruvate can take. And there are other molecules created in between this process that can also be used to make other molecules. So this slide is just to put into perspective how important glycolysis is in affecting metabolism. So if we consider obesity, which is the imbalance of energy intake and energy expenditure, we see that in a study done by Chow Dong et al., increasing glycolysis in the liver caused mice to lose weight. And this makes sense because if you're not increasing your food intake, which is where you would get glucose from, you need to go and get it from your storage. So if you use up your storage, then you lose weight. And if we consider the Warburg effect, which was proposed by a biochemist called Warburg, he proposed that cancer cells prefer to use glycolysis rather than any other generating cell. Now, glycolysis doesn't produce the most energy. There are other processes that do produce more energy, but they prefer glycolysis because they can use the building blocks made to create other molecules that are essential for their growth. And in an article done by Warburg et al., liver cancer cells are shown to have increased glycolysis compared to other normal liver cells. So what is the estrogen-related receptor? Well, it's from a family of nuclear receptors that are a class of proteins that are lignin regulated. And this means that they need a substrate in order to be activated. So if you look here, this is my substrate. And it enters into the cell. And once it's in the cell, it can interact with receptors. So in my simplified cell, we have two receptors, this one and this one. But as you can see, it would not be able to interact with this one because it's not the correct size or shape for that receptor. So it can only interact with this one. And so once they interact, they enter into the nucleus and activate whatever process it's specific for. And so most of these receptors regulate metabolism by acting as transcription factors. So what that means is that once, say, this receptor enters the cell, it would go on and initiate transcription for some protein. And so the lignin is actually unknown in fruit flies, and this is why we don't know what affects its expression. So can receptors have an effect on metabolism? Yes, they can. Receptors are incredibly important for regulating, and that's a way that your cell controls processes in the cell. So if we didn't have this regulation, we could have something like uncontrolled cell growth, which is bad, because that's essentially what cancer does. And so in a study by Olson et al., the inactivation of this receptor called alpha caused an increase in total body fat in mice. So why did I focus on the larval stage? Well, during the larval stage, which starts from the time that it becomes an embryo, or from the time it hatches from its embryo, it has a 200-fold increase. Now this is incredibly big compared to like its adult stage, where it has minimal to no growth. And so this is where we suspect it to have a lot of changing in ERR levels. So we focus on the mid-L2 and mid-L3 stages because we expect there to be a relatively high amount of ERR in the mid-L2 stage, and we expect there to be a decrease in the amount of ERR in mid-L3 stage because this is when it's stopping its growth and it's going to go into its pupa stage. So this is just an overview of the project I did and what I'm going to be talking about for the rest of this presentation. So I needed to determine whether ERR is modified, and so I had to create a 6-time his-tagged ERR PUSC construct, and I will talk about this more later, so don't be confused. I'm going to test whether ERR activity is affected by nutrient uptake, and test whether ERR is present at constant levels during larval development. So what is a 6-time his tag? Well, it stands for a polyhistidine tag, and if we break down this word, poly means many, and histidine is just an amino acid, which is a component of this tag. So my 6-time his tag just means that it's composed of 6 histidines together. And so this is just a sequence that is attached to my gene of interest. So if you're still confused about what I mean when I say a tag, you can think about a barcode on an item. So a barcode on a can of beans will be different than a barcode on a can of corn, so that when you scan it in the computer knows which one is the beans and which one is the corn. And so we have the his tag attached to our gene of interest, the gene for the his tag. And so once we create the protein, it'll have this separate structure attached to the protein. So why did I create this construct? Well, because we've seen that there appears to be a modified ERR during the late larval stages. And when I say modified, I mean that there's something attached to it, but we don't know what it is. So when they've tried extracting it in the past, it's been unsuccessful because before the extraction is done, something is breaking off this modification. So they think it might be a very efficient enzyme. So we're hoping to extract it using nickel under denaturing conditions. And this means that the enzyme will change its shape and no longer be able to break the bond. So the polymerase chain reaction is a method that I use to amplify my DNA. So we start out with the double-stranded DNA, then we heat it to approximately 95 degrees Celsius, and this causes the bonds in between the DNA to break and the strands to separate. And once they're separated, we can attach our primers to the strand, and the primers are just small sequences that are specific to whatever region we're interested in. So once they're attached, we heat it again to about 72 degrees Celsius, and this allows extension by the addition of nucleotides to it. And so how did I create my 6-time tagged construct? Well, I started with just the gene and then the his tag separately, and after I did my PCR reaction, I had the his tag attached to my gene of interest. Then I topo-cloned, which is just a way of creating many copies of this gene and into like bacterial colonies. And then I DNA isolated my interested gene from the colonies. Then I sequenced it to make sure that I had the correct gene and that it was not mutated. And then once I correctly got it, I inserted it into my vector that I was interested in, which is the PUAST vector, and it's just a vector that's specific for flies. So this is the result of my construct. This is a gel electrophoresis, and this is my ladder. And the ladder is just a bunch of DNA that's of known sizes that I can compare my construct to. And the reason I did this was because I was having difficulty at first getting my construct into my vector, and I was not sure why. So I wanted to know whether, when I was doing my isolation, if my gene, or if my construct wasn't being completely taken out, and that's why it was not being inserted into vector. But it turned out that the enzyme that's actually supposed to stick them together was bad, and so we got the new enzyme, and then it worked. And so I sent the lab off. I sent the vector off to be injected into flies, and got the strain grew up, which we can then use to extract ERR with the modification, which I didn't actually get to do because I didn't have enough time. So does nutrient uptake affect ERR levels? This was my other question. And to address this, I did laxity staining using semi-defined food versus chemically defined food. And the difference is that the semi-defined food has more yeast in it than the chemically defined food. And then I tested it with machisterone versus ethanol. And machisterone is a sterile hormone that we think is a substrate for ERR, and that it should cause expression in ERR. And then we use ethanol, which is just a control that we don't expect to see any change or effect in our expression. So we use two strains, the N-LAC-Z, which is our negative control, and the ERR-LAC-Z, which is our experimental group. So what is LAC-Z staining? Well, it's a method to observe expression of genes. And so our ERR-LAC-Z strain is a cross between the GAL4 strain and the UAS-LAC-Z strain. And so the GAL4 strain expresses GAL4 in certain tissues. So in my experimental group, it expresses it in the salivary glands and the fat bodies, and I'll let you know what that is later. And the UAS-LAC-Z strain just has this complex inside all of its cells. And so the way it works is that GAL4 will be expressed into the tissue and interact with the UAS attached to the LAC-Z. And it will cause LAC-Z to be activated and create this beta-gal enzyme. And then the beta-gal enzyme will interact with the X-gal. And so once it leaves it and creates a separate substrate, this substrate will cause blue pigmentation in the tissue. And so that's how we know beta-gal is active. So my LAC-Z staining method, I started with my egg caps, which I just collected off the top of these bottles, and they just have yeast on them, and that's where the larvae is grown and it eats the yeast. And so I collected the larvae 60 to 72 hours after, when they were at their mid-L2 larvae stage. And then I put them on agros for 2 hours, and what this does is it clears its gut from any food it's eaten off the egg caps. And then I put them on semi-defined food or chemically defined food for 4 to 6 hours. And after this experiment, I did a follow-up experiment with the machisterone or the ethanol in the semi-defined food, or the machisterone and the ethanol in the chemically defined food. So this is my results for the semi-defined food condition and the chemically defined food condition with no machisterone as yet. And this is my negative control. So here, these are the salivary glands, and these dark blue dots are the staining. So it looks blue, but because the camera is black and white, it's just black and white. And so all of the salivary glands have staining, and this is our positive control. And so these are the fat bodies, and we don't see any staining in the fat bodies for the negative control because they're the negative control, they're not supposed to have staining. There's heavy staining in the larvae with semi-defined food, and there's no staining in the larvae with chemically defined food. So then we went on to do chemically defined food with the machisterone because we thought it would actually cause expression of ERR when it's on chemically defined food. So this here is my negative control again. This is chemically defined food. This is my negative control, sorry. This is chemically defined food with machisterone and chemically defined food without machisterone. But there's no staining in the fat bodies at all. So there goes our theory that machisterone actually promotes expression. So because of these results, we decided to try it on the semi-defined food because we know semi-defined food has expression. And so here's my experimental group. Here we have semi-defined food with the maxisterone, semi-defined food without the maxisterone. And we do see that there's less expression when maxisterone is present than without maxisterone. But there's a problem with these results. Because if you look at our positive controls, they're all unevenly stained. So why could this be? Well, we thought that maybe because we had multiple people dissecting the larvae and then staining, that there was some unequal dissecting and so unequal staining between the larvae. So these results were not very good. So is ERR present at constant levels? This is my last question. The reason why we're interested in this is because if ERR is present at constant levels, and we know that ERR seems to initiate glycolysis, and if it is, like I said, present at constant levels, then what is causing ERR to not keep glycolysis going? Because we don't see any growth, or we don't see much growth during the adult stage. So there's something that's inhibiting ERR expression. And so we used fed and starved conditions as well, just to test the nutrient abundance on the protein levels itself. And we wanted to know whether total protein level is affected or whether just the ERR levels are affected. So we did a Western blot to test this, and a Western blot is a method used to identify protein by comparing it to a ladder that just has proteins of known sizes. So I started with my sample collection again off egg caps, the same way I did for my lactate staining. And then I put them on semi-defined food or agarose, and agarose is a starved condition. And I left them for 0, 6, 12, or 24 hours. And then I prepped the samples and ran them on a gel. And so the way that we did this, the gel, so this here is our gel and it's in a cassette. And these are little wells where the samples go, so it's one sample per well. And I usually load the ladder on this side and then the samples next to it. And so once the gel is run, it'll separate the proteins by size, and so we see a difference in leveling. And so we transfer proteins from the gel onto a membrane, and then we image the membrane. So here, this is an example of a membrane. So we have the protein on the membrane, so this is after it's been transferred from the gel. And then we put this membrane in a solution with a primary antibody that's specific to our target protein. And then put it in another solution that contains a secondary antibody that's specific to this primary antibody. And this secondary antibody has an enzyme on it. And so once we're ready to image it, we put this solution on it, and the substrate in the solution is cleaved by the enzyme, which causes a light reaction, and then we can image the membrane, sorry. So these are the primary antibodies I use. I use alpha flag, which is specific to ERR, so it won't bind to any other protein. And then I use beta actin, which is specific to actin proteins, and is found in all animals. And this is my loading control, so if I'm interested in looking at, or if I see that the levels of ERR have dropped, I can compare it to my beta actin and check if the total overall levels of protein have dropped or if it was just ERR. So here I have my membrane. This is the membrane with alpha flag as the primary, and this is the same membrane, but this time with beta actin as a primary antibody. And so here is where the unmodified ERR will be. And you can see that from 6 hours to 12 hours, ERR does increase. But from 12 hours to 24 hours, it doesn't really increase. It kind of looks like it decreases a little bit, and it's a lot more spared. And here is where we think the modified ERR is. And so, again, from 6 hours to 12 hours, it increases. And from 12 hours to 24 hours, it also increases. So there's an increasing in the modified ERR as the larvae gets older. And if you look at the starved conditions, there's some ERR, but not much, and we don't really see any change in it. And so this is our beta actin. You can see that it's not the best looking beta actin membrane. There's differences in the protein levels, which is not what we should be seeing. So that suggests that there could have been an issue when I was loading my gel. So my concluding remarks. The results of my lab Z staining were not very good. The salivary glands were very unevenly stained, so that's something that would need to be redone. And there's constant smearing on my Western blots, which could either be for a couple of reasons. There could be problems with my equipment, and the reason why I say this is because other people in my lab were also having issues running Western blots, or I could have had bad or uneven loading of my samples. So these are my references, and I want to take this time to thank the people in my Chem 680 class and Dr. Dave, who helped with my presentation. Questions?
So can you go back to the slide where you have the life cycle and you have the little larvae things and then the bigger ones? Go team. Oh, there we go. So I'm curious, so if you go from 1 day to, in 1 day's time, or I guess 3 days time to get from the small stage, which stage were you looking at again?
So once they become an L2 stage, we get it in the middle before, like right when they become L2 and in the middle of before they become L3.
Okay, I'm curious one, I guess 2 questions. One, how do you know that they're all at the same stage that you get them? And then what sort of variability within that group that you have? Because it seems like so much growth happens in between here. I'm curious if that variability is going to cause some trouble in analyzing some of the data later on.
Yeah, it was actually really hard for us to get it synchronized. What we had to do was keep changing egg caps, and we had to constantly come back and change them and then collect them at certain time points so that we know that whatever eggs were laid on there was laid at a specific time. So I guess what I'm trying to say is, so like I'll have an egg cap in, I'll remove that egg cap, put another egg cap in, leave it for a couple of hours so that whatever eggs are laid on there, then I would take that one out and put a new one in and keep this one. And so they grow up and then they're synchronized.
So you're pretty much like on call, like as soon
Yeah.
as they're doing their thing, it's like, oh, gotta go collect them.
Yeah. And I can't remember your second question. Oh, if it gives problems.
Right.
Yeah, it would, because then we won't have very good data with our ER expression because it changes as they grow.
Any other question? You said that this started because they isolated EIR with something attached to it.
Yeah.
Any
It was just a
ideas?
Western blot. They think it could be sumo, but they're not sure, and I don't really know what sumo is.
I was gonna say.
Yeah, they just told me that it could have been a sumo attachment, but
Should we be thinking like in terms of small molecule or other protein?
I'm not really sure.
Okay.
Sorry.
Can you just explain the difference between the ERR and the modified ERR again? And then, were they at different levels in your Western blot because the modified is lighter in weight or?
Okay, so go back to my Western blot.
Sorry.
No, it's okay. So I believe this is actually bigger.
Oh, okay.
So it's something attached to it, so it makes it a bigger protein. And so when we extract, or when we do like the gel, it comes up higher because it's bigger. And then you wanted me to explain the unmodified. I just not sure.
What's the modified mean again?
That there's something attached to it.
That there's something attached.
Yeah, so like a post-translational modification, for example. So it could be like, if you think, I'm not sure how to explain it to you, but
Like
say you have
phosphorylated or something.
yeah, so like you have a chemical and then like you have a molecule and
Yeah.
you've like added some kind of other thing to it, it becomes something different.
Yeah, okay.
So you use your ladder to figure out about how big your piece is, right?
Yeah,
So what's the spacing between?
I'm not really too sure.
Okay.
I didn't actually get that because they have a special thing in the lab for the ERR, but I never got down the numbers when I was doing it.
Sure.
So I'm not really too sure.
So were you able to quantify the expression in these Western blots? And like you said that for the top level there is like different expression, and I can see that kind of. Like, is there any way that you were able to like quantify it?
There was someone else, Anna, who was helping me with the Lassie staining, who was doing things like that, but I don't have her data because I wasn't working on that with her. So my project is just a small part of like the overall thing that they were doing. So I only know like the things that I did, so I'm not really too sure.
Okay.
Sorry.
No, that's totally fine.
Yeah.
This fascinates me. So you said that, when you said that the dissection variability, when you say dissection, do you mean with like a scalpel and like opening them?
So we actually use little tweezers.
Oh my God.
So the scalpel is too big for them, so yeah, we use little tweezers.
So I was just curious, like this, so if there's variation in the dissection, would there also be variation based on the fact that the tweezers are probably about, I mean, they gotta be the same.
Yeah.
These things can't be big. I mean, I
hard,
hope they're not big,
it's
because that's
hard
scary.
to dissect it, so we actually have to practice a bit, but how well each of us got, someone could be better than the other.
Mhm.
So
can you, do you, like roundabout numbers of these things that you put through this process?
sorry.
How many little larvae things did you actually?
So I think for each experiment we did about 20 for each condition.
Cool.
But we did Western blots many times because we kept seeing the smearing.
Mhm.
And this was actually the best we got sometimes. There was smears all over the Western blot where we couldn't even read it.
So I've never done a Western blot. Why are those, like when I compare the left and the right, I'm using a different antibody. I see the blobs, like your ladder looks like it's in the same spot, but the peaks are very different. So I guess why is the one spot in lower?
Okay, so this ladder was actually drawn with a marker, so it doesn't come up on the membrane.
Okay.
I don't really remember why that was, but it wasn't like, yeah, like when we get the marker, it's visible on the membrane. So
You don't mean like Sharpie marker?
Sharpie marker, yeah. So like we take the like photo image part and we line it up on the membrane and then we like draw it with a Sharpie.
Okay.
Bigger universities like Indiana, etc., don't have as nice equipment as we do. So ours will digitally find all of the markers on the membrane and calculate all the molecular weights for you, detect all the bands. The more primitive, smaller universities like IU, they use Sharpies and shit like that.
I'm thinking like biomarker and this and that.
No, you need marker.
No, it is a marker.
It gets the job done.
So,
It does. I used to do it that
Why are
way.
they so different? Shouldn't they be the same?
Yeah, so I'm not sure what this protein is supposed to be here, but we're really only focusing on this one. And this here is, I remember this one starts out blue, but
So I'm sorry, actually, I'm not asking about the ladder. I guess
Okay.
I'm asking about why the two, everything but the ladder seems different. Because those should be, aren't those this, like the one on the left and the one on the right, aren't those the same protein?
oh, you mean these here?
Yeah.
No, this is beta-actin and this is ERR.
But aren't you just using that to, are you measuring the antibody or are you measuring the actual protein sticking to
The
it?
protein. Okay, the antibody sticks to the protein and then it like creates a light and that's how we image it.
Okay.
But the beta-actin won't be in the same level as the ERR because they're different sizes. But the beta-actin is just like a loading control, so it's not the same as the ERR
Okay.
protein. It's a different protein, but I'm not sure.
Oh, okay. So
You're right.
So, yeah, like I said, this is not like a gotcha question, this is how does this work question. So should I think about this as like the protein you're looking for is attached to this beta-actin or something, and that beta-actin is the thing that's actually connected to the...
No, I don't think beta-actin is connected to the ERR, right? It's like a separate protein
Okay.
all on its own, but it's a protein that's present all the time, so we know that we can use it for a loading control.
So it's just the sheer size of the beta-actin that's causing the difference in size. Okay.
Yeah, so, for example, these are actually supposed to all be the same because it's supposed to be the same amount throughout the protein for the beta-actin. So like if we see here that there's less ERR compared to here, we could look at the beta-actin and see if there was less beta-actin here than here, and then we would know that there was something wrong with the way we loaded it, and not necessarily that ERR is present less than in this condition.
Sure.
Does that make more sense?
Yeah.
questions?
Let's thank our speaker again.